Hello and welcome everyone to this podcast for human genetics. In this podcast, we're going to continue our discussion on the extensions of Mendel's rules. We'll go ahead and get started here on epistasis. We can define this in a few ways, but let's first say when one gene hides, that is, the expression of a second gene. This is the first extension of Mendel's rules where we're talking about two different genes and not just two different alleles of the same gene. So these are two different genes at two separate loci. So we could think about this as a pathway where you may have reactants A that are converted into B that are then converted into C. And in this example here, you might have an enzyme 1 that facilitates the conversion of A to B, and then you might have enzyme 2 that facilitates the transformation of B to C. These enzymes are coded for by genes, so gene 1 and gene 2. So gene 1 makes enzyme 1 that functions in this pathway. Gene 2 makes enzyme 2 that functions in this pathway. So what we would say here is that gene 1 is epistatic to gene 2. And this concept, I think, becomes a little confusing. So I'm going to say this in a slightly different way. What I mean by this is that if gene 1 is gone, you never make product B. And if you don't make product B, it really doesn't matter what gene 2 is. If gene 2 is mutated or if it's not mutated, it doesn't matter. Gene 2 could be fully expressed, but if gene 1 isn't there, you'll never know it. So gene 1, if it's not present, hides the rest of the pathway. So we say gene 1 is epistatic to gene 2. This can also be confused with complete dominance, where big A is dominant to little a. This is different because complete dominance refers to a single gene that has a dominant and recessive allele. So that's not what's going on here. This is when you have two different genes that interact with each other in a common pathway. So an example of this is going back to our blood types. So let's use our ABO blood type to talk about epistasis. And we didn't go into details about this earlier, but the pathway to make A or B or O blood is more complicated than we let on. We know that in the course of making the red blood cell, that at some point you make a precursor. I'm just going to show that as a circle. We know that that precursor then, by acting with the I gene, so either IA, IB, or little I, that we can add the sugar to the red blood cell to make it type A blood. We could add the sugar B to it to make it type B blood. We could also add both A and B to make it type AB. And then we know if the genotype of the individual is little I, that you would just not have any of these sugars on the outside and it would be type O. But how do we make this precursor? And instead of calling this precursor, let's give it a new name. Let's call this substance the H substance. And I'll explain why we're going to call it that in just a moment. What we want to think about now is what makes this H substance. It turns out that there is an initial precursor. Let's just make it a triangle. And this one we'll just call a generic precursor. And we know that a gene called H makes an enzyme that converts this precursor into the H substance. We say that H is epistatic to I because it comes first in the pathway. H takes this precursor, turns it into this H substance. Now, we can't put the A, the B, or both A and B sugars onto the red blood cells unless you make H substance. So this is the initial pathway that is necessary to move forward. So let's look at a few genotypes to help understand this. If somebody is genotypically big H, little h, it could be big H, it wouldn't matter in this case, and then IA, I, little I, their phenotype would be type A. And the reason for that is that you have the H enzyme that converts the precursor into the H substance. Once the H substance is made, you have the I gene that can now take this H substance and make the A, sugar on the outside of it. If somebody was big H, and then little I, they would be type O. And that's because you have this precursor, the big H allele is present, so it's able to convert the precursor into the H substance. The H substance can't become AB or AB because you have the little I. So it's just going to stay as the H substance. And in reality, the H substance is the O type. Now, let's complicate it and say, what if somebody had this genotype? Little h, A, and let's give it a B. What is its phenotype? And if you want to hit pause and think about this for a little bit, try to come up with some possible answers. Okay, well, if you hit pause, welcome back. If you didn't hit pause, let's just move forward and talk about this. Remember, you don't have the big H. And if you don't have the big H because it's gone, right? It's now little h in this example. You can't convert the precursor into the H substance. If you can't convert the precursor into the H substance, the pathway stops here. You don't see anything beyond here, ends here. H is epistatic to I. H masks the expression of this. If you don't have H, it's done. The pathway's over. So what's the blood type then? Often, as students are learning this, they want to say it's type O. But remember, you can't be type O unless you can make the H substance. So what this individual would be is a new term, and I didn't expect you to know this unless you looked it up, but we call this type Bombay. Somebody who is little h has type Bombay blood. In this case here, if they're little h, it doesn't matter what the I gene is. It could be big A, big B, AA, BB, little I, doesn't matter, because the pathway ends here. So why do we call it Bombay? Well, this deserves at least a mention here. It's called Bombay because in Bombay, India, several decades ago, I believe it was in the 40s or 50s, a woman was in a car accident. And she was taken to the hospital. And at that time, they knew enough about blood types that they gave her type O. So you have a sick person, you gave her type O because we think of the type O as a universal donor. Anybody can receive type O. However, when they gave her type O, sadly, she died. And that's because she was little h. Her blood on the outside of it wasn't O, wasn't A, wasn't B, wasn't AB. If we use our symbol we used on the last slide, it had just this precursor. And that precursor, since it didn't have the H gene, it was little h, it didn't go any further. And so when they gave her type O, that blood was viewed by her immune system as foreign. And so her immune system attacked the type O blood and ultimately killed her. So in reality, type Bombay is really the ultimate universal donor. However, it's very, very rare. So if you're ever on Jeopardy and they ask you what the universal donor is, you probably still should say type O so you win your money. But deep inside, you'll realize that it's actually Bombay. And the next time you're at dinner with family or friends, you can wow them with what you've learned in college, and maybe they'll pick up the tab. The next thing I want to talk about are the concepts of penetrance and expressivity. First, let's define the two terms. So penetrance is the percentage of individuals with the same genotype that have the predicted phenotype. Expressivity is the degree of the phenotype's severity. A great example to describe both of these is a trait called polydactyly. Someone with polydactyly will have more than 5 digits per hand or foot. So with this example, let's talk about penetrance and expressivity. We can say a trait is completely penetrant. We would say that the trait was completely penetrant if 100% of individuals with the polydactyly, I'm just going to put PD, polydactyly genotype, were polydactyl. Meaning that everybody in a population had the genotypic mutation for polydactyly were actually phenotypically polydactyl. Then we would say a trait is incompletely penetrant if less than 100% of individuals with the polydactyly genotype were polydactyl. Now, we can often be more specific than this. So if we said 40% of the individuals with the PD genotype were polydactyly, then we could say it's 40% penetrant. So let's write that down here. Can be more specific if we know the percent of affected individuals. Like I said previously, if we know the percentage of individuals, say it's 40% of the individuals that have the PD genotype actually have polydactyly, then we would say it's 40% penetrant. What I'm saying in that example is that the other 60% of individuals with the PD genotype are actually not polydactyl. They have 5 digits per hand or foot. We don't always know why traits aren't 100% penetrant. In fact, a lot of traits aren't 100% penetrant. Most traits have some degree of being incompletely penetrant. So what about expressivity in this example? Remember, this was about the degree to which somebody was affected by a trait. So in our example of polydactyly, what we might say is that in a population, some have more than 5 fingers, and others have more than 5 toes per foot. Or you could have other variations of this. Maybe somebody is polydactyly on their left hand but not the right hand, and other people in the population are polydactyly on their right hand but not their left hand. All we're saying is that every individual who has polydactyly doesn't have the exact same phenotype. There is some variation within these individuals. All right, let's go ahead and move to our next topic of pleiotropy. I may go back and forth calling it pleiotropy or pleiotropy. Either are acceptable. Let's begin with a short definition of pleiotropy. Pleiotropy refers to a trait, or it could be a disease, caused by one gene mutation that has several phenotypes. Or you might think of those phenotypes as symptoms if we're talking about a disease. There are several examples of pleiotropic traits or diseases, but the one I want to talk about with you today is a syndrome called Marfan syndrome. Now to talk about this, let's go ahead and just draw a diagram here of a gene. This gene, which I'll just put down as big M here, when it's not mutated, it functions in making a protein. This protein, when it's made, goes on to function in the aorta, bones, particularly the bones in the fingers, the limbs, and your ribs. And it also is found in the lens. This protein in these different structures helps provide the necessary strength and flexibility of these different structures in our body. I should have defined aorta. You may not be familiar with that term. That is just the main artery that comes off of the heart. So somebody with Marfan syndrome has this same gene, except it's mutated. So I'll put that down as a little m, but I'll make a little asterisk here to indicate that it's mutated. It still makes a protein, but the protein it makes is non-functional. So it would normally go to aorta, bones, and lens, but since it's non-functional, it can't do its job there. Some of the symptoms, because of this non-functional protein, include lens dislocation, long limbs, also long, and your book uses this fun word called spindly fingers. There's also a symptom of having a caved in appearance of the chest. So I'm going to put caved in chest. The chest isn't actually caved in, but if you were to look at somebody with Marfan, it would appear that the chest was caved in. Now these symptoms I've described here are all because of the bones not having this protein to allow for that flexibility and the lens not having that protein. Now the most severe phenotype or symptom of Marfan syndrome is bursting of the aorta. Now if somebody is known to have Marfan syndrome, they will monitor this to try to lessen that, and they can try to build that up with appropriate stents, but this is the most serious phenotype. I won't ask this on the test, but sometimes it's fun facts that people like to hear. We believe that Abraham Lincoln had Marfan syndrome. All right, the next extension to Mendel's rules I want to talk about is something called phenocopy. So let's start with a simple definition here. An environmentally caused trait that occurs in a familial pattern. And when I say familial, all I really mean by that is that the trait appears that it's inherited. We should also add that a phenocopy is associated with a trait that is often caused by a mutation. The example of a phenocopy I want to talk about is deafness. We know that when a zygote forms after fertilization, that it goes through an elaborate multi-step pathway, before ultimately a newborn baby is born. Amazing graphics here, I know. You're probably super impressed. Now, during this pathway, all the different senses, sight, hearing, touch, taste, and smelling, develop. We know that as hearing develops, that there are many genes that are important. So if a gene is mutated, we know mutation of a gene. Now, you should know as you write this, I don't mean any gene, I mean a gene that is specifically related to our ability to hear. Now, sometimes when those mutations occur, it results in the child being born deaf. Alternatively, though, there can be an environmental cause of the child being born deaf. So in this example, the mother, while pregnant, is exposed to rubella, the infection that is mostly known for it causing German measles. However, if the mom is infected with this during pregnancy, the child will suffer many potential problems during development. It can even lead to the child not making it through all of development. But it can result in the child being born deaf. Now remember, in this case, no mutations in the genes necessary for us to develop hearing. So to review phenocopy, a child can be born deaf for several reasons. It can be born deaf because of a mutation to a specific gene or to an environmental effect. And this environmental effect that leads to deafness would be called a phenocopy. All right, the last extension of Mendel's rules for this podcast will be genetic heterogeneity. This is kind of the opposite of pleiotropy. Remember, pleiotropy is when you have one mutation that causes several different phenotypes. A working definition for genetic heterogeneity is that a single phenotype that can be caused by one of several genes. To talk about this extension, let's consider the trait of blindness. We know that there are hundreds of genes that, if they are mutated, can lead to blindness. So many genes, hundreds of genes in our genome, that if you mutate them, can lead to blindness or at least affect a person's vision. Vision, of course, is a very complicated sense that we have. And there's a lot of different things that could not function correctly to lead to blindness. For instance, there are many genes associated with a functioning retina. There are genes that are necessary for functioning of the optic nerve. There are genes important in the functioning of the lens. Lots of structures need to be working properly in order to have vision. If any one of these are altered, that can lead to reduced vision or even blindness. So let's write over here for blindness, any one of several hundred genes, if mutated, affects vision. And that's what we mean by genetic heterogeneity. All right, we just have one more extension to talk about for this chapter. But I think it deserves a fresh podcast, so we don't try to squeeze too much on any one podcast here. So we're going to end this podcast now, and then we'll pick up in the next podcast talking about mitochondrial DNA mutations. If you have any questions at all, please let me know. If not, I'll see you on the next podcast. Bye.